You don't even have to look them up. All right, so when we look at membranes, membranes are found inside eukaryotic. Now notice it says eukaryotic. Why do we say eukaryotic cells inside eukaryotic cells? Why not prokaryotic cells? You should know the answer to this, or I wouldn't be asking. What's the deal? Why does it say that? Why does it say organelles? Present day knowledge of membrane structure has been built up by the result of many observations and experiments. Original membrane structure proposed by Davison and Denali was a unit membrane lipid bilayer coated with protein. Well, we'll talk about that in a minute. But the question is then, why do they say membranes are also found inside eukaryotic cells? Why do they say eukaryotic cells? Aren't membranes found in prokaryotic cells?
Oh,
Sounds like, yeah.
No, it's like found only one and the other one's found like.
Go ahead, say it.
It's like the cell membrane is like on the right.
Okay, it doesn't say cell membrane. It says, look, let's read it together. All cells, see the word all cells have plasma membranes that forms the outer limit of the cell. All cells have that. So let's read, what do they mean by all cells? They mean bacteria and archaebacteria, so all prokaryotes. Fungi, plant cells, right? These three kinds of cells, the prokaryotes, the fungi, the plant cells, and by the way, the protista, some protista, they all have what? Cell walls outside of the plasma membrane. Animal cells don't, obviously. Okay, that's fine. But then they say membranes are also found inside eukaryotic cells. Yeah.
Prokaryotic cells don't have membrane bound.
Perfect. Very good. Look at the qualities, the difference between the different kinds of cells that we went over. We made a chart. I said you need to know it. It's in your notes. Prokaryotes and eukaryotes. One of the qualities of prokaryotes, which means bacteria and archaebacteria, right? Those two kingdoms of life don't have membranes inside the cell. Only eukaryotes do. Fair?
yeah.
All right. So be careful. So obviously membrane-bound organelles, membranous organelles or membrane-bound organelles are only found in eukaryotes. Present-day knowledge. Okay, so let's look at Davison and Denali. What did they suggest? They suggested initially that you had this membrane, and then outside of the membrane, stuck on it like I don't know, like bedazzling a cell, you have all these proteins. There's a couple problems with that. It doesn't satisfy how you get things through the membrane, right? They're stuck on the outside. It satisfies some of the qualities of a membrane, but doesn't explain how membrane functions. How do you get things in and out of the cell if the proteins are just sticking on the outside? So that didn't work. Today we accept the fluid mosaic model. And it's described in detail here. Here is a molecular model showing how phospholipids structurally orient themselves to form a bilayer. So let's take a look at what they mean by this. Out here are your phosphate heads. Do you see them? I'm going to go ahead and put them, make them blue. They're all kind of sticking out. You see how kind of crazy this looks? And then you have your fatty acid tail sticking out on both sides, creating this hydrophobic, right? Hydrophobic fatty acid layer in the middle, and there's a bilayer here, so you got two layers of these phospholipids. One's here, and the second one here. Two layers, bilayer. So that's what we're talking. We've drawn this. I've seen you all draw it on yesterday's quiz. I know you know what these phospholipids look like, but I just wanted you to see that this is a model that was was made by a computer using predictable algorithms. So this is not electron micrograph or anything. This is a model, another drawing, but this drawing was done by a computer based on observations and numbers and measurements, right? So let's look at a drawing. Here you have your phospholipids. Which would you rather study? Would you rather study something that looks like this or study something? Do you see how useful models can be? Right? That's why we redraw things. So there are these lipids. These are phospholipids. There's a phosphate head. There's a fatty acid tails, right? What's this thing stuck here in the middle? In between the phospholipids. What is it? Who knows what it is? Very good. Cholesterol. Excellent. This is cholesterol. I did a horrible drawing when I drew it, but there it is, cholesterol. And it says it right there, by the way, cholesterol. Some of these phospholipids have these sugars that stick off it. They're called glycolipids. Why do we call these glycolipids? Because there's a sugar on it. There's a bunch of sugar stuck on it. Here, this is a protein that crosses the membrane, right? So this is an integral membrane protein. Any protein across the membrane is called integral membrane. This is a carbohydrate stuck on it. So what do you think we call this one? It's a protein. So what would you call it? Good. Carboprotein is a good guess, but it's actually a glycoprotein. So they're called glycoproteins. So you're not making bad guesses, but it's called a glycoprotein, and this one's called a glycolipid. So there are these lipids. We said that lipids have these qualities and proteins have these qualities, but your body's able to keep making changes. Life adapts and changes and makes things takes simple things and makes them more complicated in order to do more complicated tasks. So now here's an integral protein here, but it doesn't go across the membrane. This one's a carrier, so this one actually will attach. You see how something can attach here. This is a channel protein, right? So each of these is going to do a different thing. Obviously, this can't act as a channel protein, can it? No, this is signal protein. Something's going to bind to this and send a message inside. This is a carrier. We're going to talk about carriers probably next week. This is a channel. This is going to let stuff in, right? Obviously, it's a tube. Now look at CO2. It just goes right through. Why does it go right through? Because of gas. It's a nonpolar small molecule. It dissolves in water because water can dissolve gases, so it's not a problem. CO2 is dissolved in water, goes right through the membrane. Maybe this is inside the cell and it goes out, or what have you. So that's important to understand. Gases can get through membranes. That's how you get oxygen into your blood. I mean, how do you get oxygen into your blood if gases can't pass a membrane? So you guys can read that and study it. You need to know the fluid mosaic model. We've drawn this, we've looked at it, we've labeled each part, you've been quizzed on it. So if you can't look at this and you don't know what every part of this membrane is and how it works, you have any questions, please see me. You should be able to answer these, except for this stuff. Facilitated diffusion, we're going to talk about that. Cell signaling, active transport, etc. You'll have to fill these in as we go along. Does that make sense? You'll have to come back to fill some of this stuff that we haven't covered. Some of this stuff we've covered, Ray. You can do it. But some of this stuff like facilitated diffusion and active transport, we're about to cover today and Monday. So you'll have to wait till after today and Monday to be able to answer them. Are we all everybody okay with that?
Yes, sir.
All right. So the question was, what can we do if we're taking notes? Well, if you're taking notes on this and you're drawing in your notebook, you might want to draw in your binder something that looks like this. So there's your fatty acid, right? And then you might go ahead and attach kind of a bunch of sugars onto it like this, kind of sketching it. Remember that we're sketching, so it's not the end of the world if it's a little messy, but it is important that you be able to identify what it is you're drawing, right? So it doesn't do you any good if you draw a mess and nobody can tell what it is. So these are all connected to one another, and you can, and if you have your colored pencils, you can draw them out, whatever. So you color code them, whatever you want. So this is kind of onto this lipid with its hydrophobic tails and hydrophilic head. You have these sugars, and these sugars, this is called a glycolipid. So it's half, right? Or I shouldn't call it half. It's part, somebody want to close the door, part sugar and part lipid, right? Glycolipid. What's interesting about a glycolipid is that this is what identifies your blood type. So why is it she cannot give blood to me, but I can give blood to her? Because I'm a universal donor, she's a universal acceptor, she's AB. The reason is that she has these markers. These are markers. These tell her blood, her body, her white blood cells know that it is her blood, because she has these things that stick out of her cells that are known as glycolipids. They identify her blood type as A, and she has another set that's B, so she's AB. I have none of these. I don't have anything on the outside of my cells that mark my blood, so I can give to her, because she couldn't tell since she can't tell that it's not A or B, because there's nothing there for her to read, right? Because I have nothing on the surface of my cells. If she gave blood to me, my white blood cells would register an A. Oh my God, there's something there. That's not me. What would my white blood cells do? Then reject it. They would attack them. They would attack her blood, and I'd get a fever and maybe even die. Probably die.
Because I have the wrong blood?
I'm having wrong blood. That's why when you're getting a blood transplant, it's important that you know what blood type you are. You should know what blood type you are. In fact, if you look on your iPhones, I'm sure the droids have them as well, in your emergency part of your phone, it says, what blood type are you? Yeah.
But the doctors don't just tell us that.
Huh?
Yeah, I think my mom knows.
The doctors probably do know. Some people don't even know until they're ready to have surgery or they're ready to go out and give blood. Like when you give blood, they'll blood type it. But if you've never had surgery, there's no need for it, right? But I would know. I would like to know if I were you.
I was in a program when we took our blood by ourselves.
I had surgery, but they ain't told me my blood type.
They probably told your parents.
They know. You should ask.
So,
If you're interested, you should ask. Yeah?
so what's a good blood type to have?
There's no good or bad. It's just different. If you're most people are O. So most people are O like me, that we don't have anything on the surface of our cells. So we can give to everybody else. People that are AB are what we call universal acceptors. They can take blood from everybody, but they can only give to people with AB. And so those of us that are O, we can give it to everybody because we don't have any of these markers, but we can only take from people that have O. So which is best all depends how you look at it. All right, so that's a glycolipid. That's one of the functions of glycolipids. There's many others, but that's one. Get them from both. So if she has AB, she had her A blood type from mom or dad, and she got her B blood type from mom or dad.
So do it like go for like your size or anything?
For what?
Like your size or anything? Like what do it go off of?
Blood.
Gene. It's a gene. You inherit the gene.
Okay.
So you inherit a gene that either makes an A, a B, or an O. So it's 3. O means it doesn't work. The gene doesn't work. You don't produce anything. A, B, or O.
So like,
We'll talk about it when we hit genetics, because that's a really good example for genetics. But first, let's just go on and learn about the membrane so we can get on about the osmosis and diffusion, so we can get on about the structures and how they work, okay? So cholesterol, what does it do? It helps regulate the consistency of the membrane. It keeps it fluid, right? And it says it right there. very clearly. Hopefully you get it, you understand it, and there's no issues with it. So consistency keeps it firm but fluid. All right, firm but fluid. There it is. That's what cholesterol does. It's everything is written for you. Everything is written for you. Then you have glycolipids. Glycolipids, where they're involved in what? Cell signaling and cell-to-cell recognition. So how does your cell know that it's yours? How does the liver know that it's your liver? How does your body know it's your liver? Because of these external things that stick out into the bloodstream called glycolipids, right? They stick out. Another thing that sticks out, as you can see here, are these things called glycoproteins. So here's a protein that crosses the membrane. By the way, if this is all hydrophobic here, what is this protein?
Hydrophilic.
Well, it is in part, but what is this part here?
What?
Oh, I think the wrong thing.
This has to be hydrophobic. Then what is it? What's this part of the protein that sticks out here? Hydrophilic, right? So this all here has to be hydrophilic because it's all sticking in and out of the cell. So the proteins have different domains, right? You have a hydrophobic domain, the amino acids that are hydrophobic, and here's the hydrophilic domains, and here's the hydrophilic domain. Then sticking out here, there's a what is attached to this protein? Carbohydrate. And because of the carbohydrate, we call it a glycoprotein.
The blue is the hydrophilic.
Hydrophilic, correct. Blue for water, right? Yellow for fat, right? So these carbohydrates are attached to phospholipids and they stick out. This is a glycoprotein. Again, they're involved in that whole cell signaling stuff, right? So they can cell to cell recognition. They help stabilize the membrane. Cell signaling, which is like, hey, so something could attach here, and then that could change something down here and signal the DNA to do something, or signal the cell to do something. All kinds of things could happen. Here you have a protein that is a carrier, carries something across the membrane. Something attaches here and gets brought into the cell because it attaches here. This is how a virus attaches. A virus will attach to one of these proteins, and your cell just takes it in. Ooh, let me open up this nice present. Opens up the virus and reads the DNA, and then starts making more and more copies of this. So the virus is not really, that's why a lot of people don't argue, the virus is not really alive. It doesn't do anything. The cell does everything. The virus is just a little message in a bottle. And then your cell picks up that message, reads it, and starts to make copies. Makes more bottles and more messages. Yeah. Say that again? It's like a mailbox in a way, yeah.
So the carrier does that?
No, well, these receptors, the carrier does the carrier. These are general functions, right? So these are many different kinds of carriers do all kinds of things. We're going to look at one when we do active transport next week. Yeah.
So the little spiral thing is a glycoprotein?
Say that again.
The little spiral thing is a glycoprotein?
Yeah, this would be a glycoprotein, but the reason this is just an example. They don't have to be spiral, right?
I know.
But this is a protein. It crosses, and look here. Why is it a glycoprotein? Because there's some sugars attached to it. So we call it glyco for sugar and protein because it's a protein, amino acids put together. You see why it's important you know your biomolecules, right? Yeah.
I thought when sugar was attached to it, it was
Very good. Excellent. Right. Good. Listen and think. See, this is the kind of question we got to ask. Excellent question. Thank you. So when we attach, now listen carefully and pay attention. When we attach the sugar to a lipid, right? This is a phospholipid. We call it a what? Glycolipid. Correct. Good. But now, this is a protein, not a lipid. This is not a long chain of carbons. This is a long chain of amino acids, right?
So glycolipids are a long chain of amino acids.
It's a protein, so it's not glycolipid. No, two different things. A glycolipid, look at it, is a lipid, right? Phosphate, fatty acids, right? And what's attached to it? Sugar. So glycolipid. We got it? That's where now here, this is not a lipid. This is a what? This is a protein. But we still have sugar connected to it, so we call it a glycoprotein. Similar functions. This just one's a lipid, the other one's a protein. That's all.
So what's the first few words?
Glyco means sugar.
Cause glyco.
That glyco just means sugar, like glucose.
So when you draw it, it don't have to be spiral.
No, I don't care if you know it's spiral or not. I mean, that's just a drawing. That's just an example. It could be. There's all kinds of shapes it can be, but that's the drawing that they have. So whatever you want. Like you wouldn't know if this was a lipid or a protein or what have you unless somebody told you it was a protein, right? All right. Then we've seen this before because we've drawn it. This is a channel protein. So it's a protein that crosses. Remember, the proteins act like doors or windows. This is like a door, the membrane that allows things to come in and out. Now, interestingly, water is a small molecule. It's small and polar. So, interestingly, it can actually fit in between the membrane, just barely, but it does. It's polar, so it doesn't like to go through. It's hard for it to go through, but it does go through. We do have pores that allow it to move in and out. And before we start talking about facilitated diffusion, active transport, and all that, we need to understand what diffusion is. Don't you agree?
Yes.
We need to understand what that is before. So we'll come back to this. Listen to what I'm saying. We're going to come back to this, right? So don't think we're gonna move on, but I don't want you to panic and say, "Oh my God, I didn't write it down." I didn't go. Well, we'll be back to it in just a second. So to be clear, you should be able to answer C and D. Obviously we need to talk about A and B still. You definitely should be able to answer number 2 on your own, and you should be able to answer 3 at this point. You should be able to answer those. If you need help, come and see me after school or help each other out, but you should be able to fill this out. It says, let me reiterate. It says phospholipids contribute to how do the properties of phospholipids contribute to their forming structural framework of membranes? Yeah.
Is it because in some parts it allow water to travel through, in other parts it doesn't?
Phospholipids, but how are phospholipids, the properties of phospholipids? What is the property of phospholipids that we're concerned with?
Has a polar head,
That's right. The fact it has a what? A polar head.
non-protein.
It's polar head and nonpolar tail. Allow for interaction of the nonpolar ends and the polar heads. The polar heads point toward the water in
Stop that and be quiet.
and out of the cell. Creating a barrier. Also, proteins can interact and embed themselves
I don't got room for that, my brother.
Create room then, dude.
in the membrane. Anything like that where you're looking at how phospholipids having polar and nonpolar ends can interact with each other, they create a barrier, they allow proteins and yeah. Huh? Do I have a highlighter for you?
She said that she borrowed a highlighter.
So anything where you're looking at the idea that these proteins can form channels and cholesterol can stabilize or rather allow the membrane to be more fluid, that all this lends itself to allow for a barrier to occur between inside and out. We went over this, went over it clearly, defined what is a membrane, what use is it that the phospholipid has a polar head and a nonpolar tail. Why is that important? And we discussed that in detail. This is kind of a short summary of that. And I'm sure there's a way to write it in even shorter than that, but that's what I wrote. Describe the fluid mosaic model of membrane structure. And that's just simply that the membrane is constantly moving, that it's made mostly of phospholipids, but it has proteins embedded in it, like islands floating in a sea of phospholipids, that the phospholipids flip and twist and turn. This is not, the mosaic part means that it's made up of different components. It's not just made of fat or not just made of protein. It's made of a bunch of different components, as you can see here, to form a big picture, right? So that would be the idea behind that. Why is it that you could not have, why is it that we have to talk about a fluid mosaic model? Why can't it be more of a shell of protein around a lipid bilayer? Why can't that be the case? Why can't there be just phospholipids inside a protein? Why does there have to be? That's the question they're asking you, right? Fluid mosaic model. Why do you first, let's talk about the fluidity of it, or the mosaic of it. Why can't it be two distinct sections? Protein section and lipid section. Yeah. That's excellent. That's right. We need doors and windows into the cell. This model where you have protein here, and fat here doesn't work. It doesn't work. We know how much protein's in a cell. We know how much fat is in a cell. To say that all the protein's together in one section, all the fat in the other, doesn't work. Because that's not, a cell won't function because there's no doors, no windows, no way of signaling, no way of communicating inside and out. All the cells have to communicate. We literally just got done talking before we started recording this, and how your cells, how a woman's cells, her eggs, are able to communicate with her body and signal all these changes that occur. How do you signal these changes? You have to be able to communicate between one cell and the next. You still have a barrier, but you have to have ways of communicating. This leaves you no ability to communicate from inside and out. That's number one. So there you need to have a layer of lipids that have embedded in them some type of protein, and it can't be consistent. It can't be stable. It can't be fixed. The proteins can't be in a specific place every time. Why? Someone just mentioned it, and I'm sure they didn't come out in the audio recording here, so let me say it clearly. Just like in the example of insulin and how insulin works, when you think about it, what does insulin do? It changes the number and the quantity of channels, the quantity of glucose channels in the membrane. So by definition, you can't have a static, you can't have a non-changing membrane. It has to be something that can change depending on your need. So if you need to get sugar out of your blood, you have to put more channels in. If you have to keep sugar in your blood, you have to take some of those channels out. So you're constantly changing. Don't worry about it. To my knowledge, this class is not over. So you have to constantly change this membrane depending on the needs of the cell. So that leads us to why you need, so all that in some way you have to put in your own words on what a fluid mosaic model is and why is it important that number one, it be able to change, the fluidity of it is important. Obviously, you don't want it to be static, you don't want it to be stiff, you want it to be able to be flexible. Here's another thing about flexibility, your cell's swelling without bursting, right? Things that you want it to be able to be flexible to some extent. Explain the importance of each of the cellular functions. High membrane surface area, high surface area. How does that impact? How does surface area impact anything? What is that going to do? Say that again? Sort of. What is it? What are the things that impact osmosis? How do we impact osmosis? How fast does osmosis occur? Or diffusion for that matter? How do we get stuff in and out of the cell? You're breathing oxygen. And oxygen diffuses in out of the air onto your hemoglobin. It gets fixed to the hemoglobin. Then your cells release it into the blood. Carbon dioxide is released into the blood and then exhaled out of your lungs. How do we get this carbon dioxide and oxygen across the cell membrane? By diffusion. So how is surface area, does surface area increase or decrease? Do you remember Fick's law? What are the factors that impact diffusion and osmosis? So here's an example. If I have this space here, and this is the one opening that I have 20, you know what's a good example of this? Our fire drill we just had. Who saw all those people trying to get back in this building? Now think of the door to try to get back in. Think of that door as being surface area, right? That's the surface area of the cell that allows it to get through. What could you do to increase the rate of movement of students from outside the building to inside the building?
Make it bigger.
Make it bigger. So the greater your surface area, the faster and more diffusion and osmosis you get. So that's why living systems do this. Look at it. Versus this. In the same space. Do you see that? The same space. Let's call this X. The same amount of distance from here to here. Let me do it in blue. From here to here. Which side has more surface area? A or B? Which side has more surface area? A or B? A. Do you see why? What is surface area? The surface length times width. So when you're looking at this, that's a lot more surface area because I folded it, right? A lot more surface area than you have here in the same length. The length is the same here. With the distance, the room you have to fit it in is the same, but you're able to fold it up and put a lot more in the same space. Is that clear? Mm-hmm. So if you eat a turkey and you have fat, sugar, protein sitting here in your intestines, would you rather have this to absorb it, or would you rather have this to absorb it?
A.
You want to have A. And in fact, that's what your intestines do. They fold in. Each of your cells and your epithelial cells also fold in. When you look at these cells under the microscope this week, you're going to see these folds up and down on the cell membranes. The reason they're there is they increase surface area. If we unraveled who's watched those zombie movies? When the zombies eat the human, a human or even a horse and any mammal's intestines, what do you notice about those intestines? they're wrinkled, they're wrapped up, and there's a lot of it. There's many, many feet of intestine wrapped up inside your abdomen, large and small, many, many feet. If we opened you up and strung it out, it'd be very long intestines. When people do autopsies, that's what you do. When you slaughter a pig, this is what happens. You have intestine that's all wrapped up in that abdomen. The reason is you can fit more surface area. There's more of it in a small space. So folding is a big advantage to life. So you want the importance of high surface areas, you get more osmosis, more diffusion, more space to do the business of the cell. If you need oxygen, do you want to take too long? You think you want to wait for that oxygen? Do you need it right away? You need it right away. So you need to have fast diffusion, larger surface area. Channel proteins and carrier proteins in the plasma membrane. What's important about those? They're going to do what? What are they going to do? That's right, they carry stuff. But why are those important for the cell membrane? Say that again. Right, sugar is one example. Correct. Excellent. Because I know we just used the last sugar as an example a minute ago. But think about it more generally. What is important? What is the quality of sugar that sugar shares with a lot of stuff? Why? When do you need channels? When do you need carriers? When there's two times. You have your membrane. When do you need a carrier or a channel? Yeah. Vitamins is yes, but a more general. Give me a general rule. When I'm looking at anything, X, Y, Z, H, whatever it is, how do I know I need a channel for it or a carrier instead of it just... They're too big? Correct. Size? So when things are very big, and when things are what? When things are charged. If they have a charge, and they're big, or they're extremely polar, right? This makes it hard to cross the membrane. So what do you need? A carrier or a channel. You need to facilitate diffusion. Is that clear? Guys, you have to know this. I guarantee you're going to see it in May at the end of the course exam. I guarantee in March or whenever they give you this exam, you're going to see these questions. They're going to give you a bunch of molecules and say, does it cross the membrane or not? Well, is it big? Is it charged? Are they large proteins? Will oxygen cross the membrane? Sure, the oxygen will do diffusion. It doesn't need a carrier. It doesn't need a channel because it's small and it's nonpolar. But if it's big or polar or charged, guys, it needs a carrier. Am I clear on that? Yes. You should know that by now. That should be something second nature by now. If it's not, you have some work to do. This is a warning. This is you being reflective right now. Do I understand this? If not, I better come to tutoring. I better figure this out. It's actually pretty easy. Now there's another time you need a carrier. Even if it does diffuse, you might need to pump things out, and that's when you want to create a concentration gradient. All right, when you want to create a high and a low. And we're going to look at that very soon, where we're going to create high and lows. So again, you need a carrier to do that. You need to be able to pump things out. Remember the example we used in class was if you were in a little rowboat in the ocean and you wanted to make sure you kept the high on one side, naturally the water is going to go from outside where it's a lot of water to your boat where there's a little water, right? But what you need to do is you need to use energy to pump that water out or your boat's going to sink, right? So you need to be able to pump water out. But to pump it out, you need some kind of protein, some kind of carrier, something that's going to move that stuff against the concentration gradient. All right. Non-polar lipid-soluble molecules diffuse more rapidly through membranes than polar lipid-soluble molecules. Do you see that? The non-polar diffuse easier than the polar. Does that make sense to you?
Yes.
Okay. That makes sense because you can't get, if it's lipid insoluble, if it's polar and it does not dissolve in lipids, then it can't get through the membrane. I mean, I think that should be simple. That's why that answers letter B right above it, doesn't it? Literally, this statement answers B. Why do you need channels and carriers? Because you need things that are you need to be able to carry lipid, non-polar stuff that's not soluble through membranes. Why? Because of polar and non-polar. Like dissolves like. It's just that easy. Discuss the implications of transport substance through a cell membrane through the cell. And we just discussed it. I literally just told you. You need to be able to move this stuff across the membrane somehow. So you need a carrier or a channel. Or you need to do phagocytosis or pinocytosis. And we'll talk about that in a minute. Describe the purpose of cholesterol in a plasma membrane. It spreads out the molecules. It increases the fluidity. It increases the ability of the membrane to move around. Again, very important. List 3 substances that need to be transported into all kinds of animal cells in order for them to survive. What are 3 substances that have to be transported? Oxygen. All right, oxygen, I'll say yes. I'm going to say yes. But let's think about things that need to be transported. Sugar. Like you have to actively go and find a way across. Like oxygen will diffuse. I agree with you. Oxygen is one of those things and it happens through diffusion. Okay. But what about stuff like, okay, sugar, carbohydrates, large things, right? What else? Blood? Blood's a solution. It's not a substance. I mean, it is a substance, but it's a solution. And the cells are in the blood, right? And the blood touches the cells, but the blood doesn't actually go into cells. Cells are much smaller than blood. So, I heard sugar. What else? Proteins. What else? Lipids, right? Gotta get all 3 in there. List 2 substances that need to be transported out of animal cells in order for them to survive. And you know what? Why don't we go ahead and add oxygen, right? Sugar. What was the other one we said? And you can pick anything, really. There's so many different ones you can pick. Proteins. And I picked oxygen for a reason because I looked at the next question. I said, okay, well, let's pick something else. What would you need to get out? Carbon dioxide, correct. Do you see why I picked oxygen? CO2, gas, right? This is gas. Now, those will diffuse, and what's the other one? What do you need to get out? Nitrogen waste. We call it what? Well, it's not poop, actually. It's urea. Poop is stuff that never was in your body. Poop was never in your body. Are we clear? If you have stuff in your poop that was in your body, you got a problem. You have a hole in your body. You have an ulcer. You're getting. That's when you get blood in your stool. That's no good. All right. Your poop was never in your body. All right, have a good one.
It's in your body.
No, it's not in your body.
So where's that?
It's on the outside of your. So you touch your finger, move your finger. Here, let me say this. If I gave you a ball of string, it was this big. It's a long string, and I held on to one side, tied it to a doorknob, and I told you to swallow this ball. What would happen to that ball?
Stay there.
No, you swallow it, you go down your throat, in your stomach, through the small intestine, wrap around into the large intestine, then where?
Doodle it? what? I don't know.
Well, where did we go? Where'd the ball go? I got the other end over here. Now look at me. Pay attention. Where would that ball go? Because it goes through the large intestine, and where does it go? Out your butt. So now, but look at me. Look at me. So you can floss yourself. Right? Like this, like you floss your teeth, right? So listen, think about what that means. Just think about it for a second. So that means that this hole, as you call your mouth, is one tube. This is one side of the opening of a tube. Where's the other end of the tube? So that's all outside.
Yeah.
All that's outside. You swallow something, it gets absorbed into your body where?
Wait,
In your intestines,
so
through
basically,
your cell membrane.
so basically you saying that your mouth, like, oh, I get what you mean.
You are designed as a tube within a tube.
Yeah, I get what you mean. So
Right.
basically, since it's like a tunnel, like the tunnel isn't in the mountain, it's like still outside the mountain, the mountain is over the tunnel.
Exactly right.
So what about the corn?
What about corn?
Can't corn not be?
You should not write her no pass, cause you gave her, you.
The outside of the corn is not digested. So the shell, the cellulose part, the part that's the same as this wood, it's yellow, right? That's the part we see. It's made of wood. It's made of it's made of cellulose, right? So we don't digest cellulose. We can't digest it. So we're going to poop it out. But because it's yellow and it keeps its shape, that's what comes out of our butt. But you understand, we ate the starch. Our body was able to digest and break down the starch that was inside the corn kernel. So we ate corn. Eating corn, you do digest it. It's just you don't digest the outside of the corn. You need higher surface area. Stomach. Intestines, really. But yes, good. Your stomach, your mouth. Okay, your mouth is a good answer as well. Where else would you need? Where else would you need high surface area? Well, why would you need it in your throat, though? Say again?
Why would you want food to go down faster?
Well, it wouldn't really go down faster, right? It's another story, but think about it. Wherever you're absorbing, right? Wherever you're absorbing, what does absorbing mean in for cells? Wherever you're doing what?
Something in, or diffusion.
Diffusion, osmosis, right? Wherever you're doing any kind of taking in. Anytime you're taking something in, where are you taking stuff in? Where are you taking stuff in? You already said that your skin, does your skin really, is it used to absorb things? Can you absorb things to your skin?
Yeah.
So when you're wanting to drink water, you just stick your hand in the water and drink that way? No. Eventually it will happen, but it's not made for that, right? It's not made for that. Your skin is not made for absorption. So the outside, your skin is made as a barrier. It's made to stop things from being absorbed.
So when you put lotion on, it don't absorb.
The lotion is just making the dry skin not so dry, so that you don't get cracks in the skin. Because you get cracks in the skin, then things can get absorbed. You don't want cracks, because cracks are going to open up your skin and allow things to get into your body. You don't want that. You don't want things on your skin to get into your body. So eczema, my nephew and my brother have eczema. That does happen. That causes problems. You can get infections that way, right? So you have to be careful. You have to keep it. You have to follow directions by whatever your doctor says. Okay, but you want to reduce that eczema as best you can and keep it as whatever. I don't know. You have to talk to your dermatologist. But my bottom line is that skin is not made for absorption. We did say, I heard intestines. That's correct. I'm not sure. So let me ask. I'll ask again. Somebody said, let's draw this out. Let's draw this. Let's draw a schematic. And what I mean by that is we're not going to worry about details. We're going to draw out a pattern that's meant to represent certain parts of your body.
Okay.
So let's go ahead and draw this and call this the mouth. All right, so there's no teeth, there's no tongue, there's no face. Get it? Fine. But then we have a throat, correct? So that's your mouth, this is your throat. In fact, let's call it, this is me giving you again a warning while you're being recorded. I don't know why you do this when I tell you you're being recorded. I'm asking you to focus your mind. So esophagus, we have the esophagus, that's the tube that goes down into your stomach, all right?
Is that? No.
And that's your stomach. But then you have, food goes, the stomach really, it does absorb some things, but a lot of the stomach is actually part of digestion, helps break things down. So really you have acids and enzymes, and these enzymes and these acids, what they do is they help break down food. Right? Are we clear? So that's really the essence of the stomach. The stomach is just for breaking down mostly. But then you go into the intestines. Now we start to absorb. Now we start to take things into the body. This is outside. Do you see this?
Stomach.
Where it say breakdown, that's the stomach, right?
Yes, ma'am. I said again, this is schematic, which means that we're not caring about detail right now. We're just looking at placement.
Perfect.
So if I took and I made everything that's outside green, and I looked, so this is all green. Green, it would be green in here. It's green inside the tube, correct? It's green in the stomach. So this is all, when something's in the stomach, it's actually continuous with the outside. So stuff that's in your stomach is still outside. Are we clear? So if we were going to draw the rest of your body, again, as a schematic, we would draw something that looks like this. And I'll finish the drawing in a minute. So that's the rest of your body. This is all outside here. This is all outside. Everything outside your mouth, right? The inside here, though, this mouth is an opening. It's all continuous. It goes into the stomach, which is still outside the body, technically.
Connected to your mouth.
It's all connected. It's a tube. You're a tube within a tube.
To my tube outside.
This mouth, that so many of you like to open, is all outside. See here, right into the mouth, right into the stomach. So now, now we're going to start absorbing. Now we're going to start taking stuff into the body. So what are you going to do? You're going to absorb. So what are you going to do? What's your body going to do?
Eat.
Yes, that's right. All this is part of eating or digestion. I'll say again. What does your body do in order to increase surface area? Fold is correct.
Your body folds.
So your body folds over and over and over and over and over and over. In fact, you've all seen zombie movies, so you know, if you haven't, that's fine.
Oh yeah,
If you've watched,
intestines be like.
the intestines are very long. They're very long. They get folded around. They get folded upon fold upon fold. They get bent. So when you eat something, it literally goes in here, gets broken down. They just keeps going round and 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 round. All the tube is on the outside.
Like your stomach up.
Intestines, all that. If you think about it, it makes perfect sense. Who thinks they want poop in their blood? Do you want bacteria? When you poop out, you know most of that's bacteria, right?
Oh, I thought it was food.
It's food. Wow. I'm gonna say.
Yes, my fault.
So most of poop is back. Keep talking. I don't know what else to say. You're not focused, and that's okay. But don't. I'm saying to the class, I need you to be quiet and focus. Thank you. So all this stuff that I'm telling you, if you think about it, it only makes sense that this is all outside your body, because if it wasn't, then you would be having poop inside your body. Poop is food that you are not digested, that you can't absorb. You cannot absorb it for whatever reason. And bacteria. The bacteria that love the food you can't eat because they can eat it. So they eat it and they multiply, and there's billions of them and they love it. And you're okay with that because they're still outside your body. They're not in your body. They're outside your body.
Question.
So, wait, before we go on with questions. So it folds and folds and folds and folds. Then it goes to the large intestine. That's called the colon. There's something like this. And then what happens? Then it goes outside again. You have another hole, another opening. That's right. So now that is, all right, come on. So this is all outside, right? All this is outside. All this stuff is outside the body. Now the stuff that's blue is inside your body. Let's make it red so it's like blood then. This is all inside your body. Here's your lungs. There's your liver. There's your muscles, your blood vessels. All this is surrounding and on the inside of your body. Your brain, your heart, your kidneys, your bladder. This is all on the inside of your body. If you think about it, this is the only way this makes sense. It's the only way that it makes sense that you can have poop and still be alive. If you get bacteria in your blood, you're dead. You'll get sepsis.
That's why when people say, like, if you hold your poop, it's bad.
So that's another story. But it is bad for you, but it's not the way you think. It could cause a problem the way you think, but generally it just means other issues. But so in any case, for this reason, a lot of some people call it your design, the design of your body is a tube within a tube. We can even simplify this drawing a little more and just draw a tube, forget the folds, and then draw another tube. That's horrible.
Oh, we got so many tubes. Like, is there a free space?
What is that?
Yeah.
What's it called again? Where you said it's not about the details.
It's a schematic.
It's a schematic.
I don't know what he's telling me. I didn't know food was outside. I mean, is that what he said?
No.
I didn't know food was outside the body.
So if you look at, if we just went down and looked at you as just a worm, right? Because worms have very much the same exact body design.
For real?
Yeah, worms are, I mean, in a sense, they're very similar in body design because worms are tubes within a tube, right? So you have blood inside you, just like the worm does, just like most creatures that are, worms are not vertebrates, but their vertebrates are. So everything that's red is inside you. Everything that's green is outside the body. So this is all outside. This is your skin on the outside, your head, all the parts of the outside that are touching the outside. And then this is still outside your body. So another way to describe it is this. If I took a ball of string, really super long, this is not possible. So let's not try it. It is not possible, so let's not try it. Pay attention. But if you took a ball of string and you tied it off to a doorknob and then swallowed the ball and somehow was able to unravel, it would come in through your mouth, wrap around all the intestines, and come out the other end. And you could floss yourself, right? Because it's all continuous.
If you swallow a ball,
What'd I just say? What'd I say? The theoretical part here that I'm trying to get to you is the theoretical part that I'm trying to get through to you is that you are a tube within a tube.
Is that why when you eat noodles, like sometimes the noodle don't go all the way down, like it'll stay right
No,
there?
that has nothing to do with that. Just pay attention. So when you swallow things, you break it down. You need to absorb your food through here. Now there's another tube. You're not just one tube. There's a second tube. Be quiet. That's next to your, right, actually, they both come off of your mouth. So one tube is your esophagus. The other tube is what? One tube is your esophagus.
Your kidneys, or you're going to your lungs.
Lungs, that's right. One tube goes to your stomach and your intestines, and the other tube has to go to your lungs. Now, how many lungs do you have?
2.
You have 2 lungs. So how many tubes are you gonna? How many times is it gonna split? At least into 2. So we call this the trachea. It actually sits in front of the esophagus, but I can't draw it that way. So I'm just gonna draw it this way. It splits. One goes right and goes here to the lungs. Another one goes left, goes to the lungs.
So then what?
Redundancy, increased surface area and redundancy. Redundancy means Redundancy means that you have more. For instance, you have 2 kidneys. Yes. If one kidney goes,
I'm about to say,
you have a backup. That's redundancy.
now what if you don't
So
got
you
both?
can live with the 1 lung. You won't be able to do as much as you could with 2, but the bottom line is you can live with it.
What if you had? So what if you don't got no kidneys? You dead?
You're dying. You can go through dialysis and keep your life for a while, but you'll probably get a kidney transplant or die.
What if you had 3 kidneys?
Not likely. I don't know of anyone who does, but we'll move on. So this is also part of the outside. As you said, you're breathing because it comes from the same mouth, right? And this is all. But here at the lungs is where the outside and inside meet. So you have blood vessels all throughout here at the lung. And this is where the outside and the inside meet. The same thing goes with your intestines. That's the blood vessels. You're full of intestines. So that's why somebody gets shot in the gut. And so it can cause so much trouble because you have a lot of blood vessels there to help you do what? Bring the food in from outside. That's what this is. That's what digestion is. You're bringing the food into your body from the outside. That is absorption.
These people keep back on yoga.
So let's be clear. Let's zoom in. Here at the lungs, the splitting keeps happening.
We can hear you.
Oh, yeah. All right, thank you. Bye. All right. So when we're talking about the lungs now, think about this and listen carefully. The lungs keep branching and branching and branching and branching and branching. They keep splitting. They keep splitting. It's a good question. They keep splitting and splitting and splitting. So you see how this trachea split once here? But it keeps splitting into smaller and smaller tubes. At the very end of the tube, there's a little tiny tube with a little sac. This is called the alveoli. So at the very end, so you know there's something called bronchioles. The bronchioles are a split in the tube that has a bunch of alveoli.
That's what bronchitis.
That's right.
So
So your bronchioles are the tubes that are they have a bunch of alveoli in them, little sacs. The alveoli are the sacs. The sacs are going to fill with what? In the lung. Air. They're going to fill with air. So this is air in here. This is oxygen and nitrogen and carbon dioxide. Wrapping around the lung, wrapping around the alveoli is blood vessels.
and if those
So bronchitis is basically when your bronchiole.
They get an infection. That's right. That's it. You got it.
can you die?
It's a long story. Mucus helps keep the lungs functioning. If mucus gets too thick, it can stop the lungs from functioning. So it's a slightly of course you can. It's called pneumonia. All right, so,
Bronchitis is pneumonia?
no, it can develop a pneumonia. So now, so if we took a look at this and zoomed in, let's take a zoom in, look at this. What you would see is the alveoli, because you have so many branches, tiny branch after tiny branch, you have a lot of surface area. So, oh, I didn't want that. Stop. All right. What you have a lot of surface area. So when you have oxygen gas, can do what? Oxygen is a gas. It's nonpolar. It's small. What's it going to do? What's it going to do? This is oxygen higher or lower in the blood at the lungs? High. High oxygen is going to move to where? To low. You go from high to low. Now, is CO2 higher in the air or in your blood when it gets to the lungs? In the blood when you get to the lungs. So CO2 is high here. So where's the CO2 going to go? It's going to go out. That's breathing. That's breathing. Oxygen comes in because of high to low. This is diffusion. At the lungs, you have simple diffusion. Oxygen goes in, and carbon dioxide goes out. Is that clear? Now, that's simple diffusion. Now, when we're talking about your intestines, now we have all those folds that help with surface area, right? Because now instead of just being like a worm where you have a straight tube, you have all those folds, so it takes longer for the food to move through, so you have more time to help break down that food and absorb it. But your cell does even more than, your tissues do even more than that. You have what are called diverticula. Your intestines do this. Your intestines do this. Do you see this? They twist and turn. They fold. We call these, and so have you ever heard of diverticulitis?
No. Yeah. That's a big word.
Some of your older parents or uncles or grandparents might have it.
My great grandma.
They might have to go to the bathroom a lot. They have a lot of issues.
That's what that is.
No, not always. There's one possible. Don't put anything away yet. Don't put anything away. Just wait. Now, this diverticulitis, or this diverticula, what they do is do what? They increase surface area. Then when you look at the cells of the intestine, this is what they look like. Now we're going to zoom in again. We're going to zoom in again. And when we look at that, the cells do this. Pay attention.
When you say pay attention, that means pay attention.
Here's what the cells do. This is a cell. On this side of the cell, it does. And then this is what it looks like on one side of the cell. And on this side, you have a blood vessel, capillary.
Capillary?
So the blood is here, right?
Is that like a tube? Or like
It's just a blood vessel, another tube. You're made of a bunch of tubes. You're going in every direction. So here's your nucleus, nucleolus, endoplasmic reticulum, rough and smooth, your Golgi body. Don't worry about it. Your mitochondria, your all kinds of things, ribosomes. But what's happening here? What's happening here? Listen, think. Before you go anywhere, think. You've digested the food. You've broken it down to tiny pieces, amino acids, proteins, amino acids and proteins and lipids and fats. So you got them the tiny little pieces. How are you going to get them from here sit down, young lady. Sit down. Sit down. Stop putting things away. Sit down. You're going to disrespect me in this way. What you're going to do is you're going to get a detention. You're going to sit down in your assigned seat facing forward, and when I dismiss, you can go. Sit in your assigned seat facing forward, and I'll be documenting this behavior and writing this up as a detention. And next time it'll be a referral, the next time you do this. It's not acceptable behavior. So food gets absorbed through the membrane through this high surface area. All right, have a good day.
Thank you, Mendoza.
You're welcome.